The next chapter deals with the external morphology, that is the structure and functions of integument and molding. And coming to the structure and function of integument, the integument is outer covering of an insect and it is also called as exoskeleton. Insect body wall is called integument or exoskeleton. It is external covering of the body which is ectodermal in origin. So there will be, it's a triploblastic organism having three layers, that is ectoderm, mesoderm and endoderm. Here the integument is ectodermal in origin. It is rigid, sometimes it will be flexible. It is lighter, stronger and variously modified in different body parts to suit different modes of life of insects. Coming to the structure of the body wall consists of an inner cellular layer, which is a living layer. It is made up of cells and this is called epidermis, an outer non-cellular layer and is called cuticle. Coming to epidermis, it's an inner cellular layer resting on basement membrane, and the functions of epidermis is the secretion of the cuticle. So cuticle is secreted by the cellular epidermis and digestion and absorption of whole cuticle, and whenever the size of the insects increase, the growth takes place, the old cuticle will be replaced by new cuticle. At that time the epidermis helps in digestion and absorption of whole cuticle. Next one is wound repairing. Whenever wounds happen on the surface body of the insect, the epidermis will help in repairing of the wounds and it will also give a surface look of the exoskeleton. And coming to the non-cellular layer, the cuticle, it's a non-cellular layer comprising of three sublayers. The endocuticle, it is the innermost and the thickest layer. This layer is made up of chitin and arthropodin. This layer is colorless, soft and flexible. And next one is exoskeleton, outer layer much thicker with a composition of chitin and sclerotin. This layer is dark in color and rigid. Endocuticle and exocuticle put together are called as procuticle. And coming to the epicuticle, outermost layer which is very thin layer, 4 canals present in the exocuticle helps in deposition of cuticle. This layer is differentiated in the following layers: the inner epicuticle, outer epicuticle, the cuticle layer, the wax layer and the cement layer. The inner epicuticle consists of wax filaments. The outer epicuticle, it makes the contact with the cuticulin, and cuticulin is a non-chitinous polymerized lipoprotein layer. It's a lipoprotein layer which is not having any chitin. The next is the wax layer. It contains closely packed wax molecules, prevents desiccation. Then cement layer, this outermost layer formed by lipid and tan proteins. It protects the wax layer. So this is the diagram showing the structure of the integument. So we can see the epidermis, the endocuticle, the exocuticle and the epicuticle, the different three layers. Then the 4 canal opening of the duct of dermal gland, setae, different structures of setae, the tromogen cells, the modification of the epidermis, the trichogen cells, dermal gland, etc. and also the basement layer from which the epidermis is laid out. Then composition of the cuticle. The cuticle is having chitin. It's the main constituent of cuticle, which is nitrogenous polysaccharide and polymer of N-acyl acetyl glucosamine. And chitin is water insoluble and soluble in dilute acids, alkalines and organic solutions, solvents. And the next component is arthropodin, an untanned protein which is water soluble. Sclerotin is a tanned protein which is water soluble. Resilin, an elastic protein responsible for flexibility of wing sclerites. And coming to the endoskeleton, cuticular ingrowths of the body wall providing space for muscle attachment is known as the endoskeleton. There are two types of endoskeleton, the apoderm and the apophysis. The apoderm, hollow invagination of the body wall, ridge-like. This will be ridge-like. Apophysis, solid invagination of the body wall, it will be spine-like. These are the two different types of the ingrowth of the endoskeleton, the apoderm and the apophysis. We can see the apoderm and the apophysis in this diagram. Then coming to the cuticular appendages, non-cellular appendages have no epidermal association, but it is rigidly attached. Examples: minute hairs and thorns. And cellular appendages have epidermal association. It may be unicellular or multicellular. Coming to unicellular structures, the unicellular structures are clothing hairs, plumose hairs, which are seen in honeybees, and bristles, which can be seen in flies. Different types of flies will be having bristles. Scales are flattened outgrowth of body walls, which are seen in moths and butterflies. The glandular setae, which are seen on the Lepidopteran caterpillar. Sensory setae associate sensory neurons. Setae are hair-like outgrowths from the epidermis. Epidermal cells generating setae is known as trichogen. The epidermal cell which forms the hair, that is setae, is called a trichogen, while the socket forming the cell housing the trichogen is called a stromogen. In both sides of the trichogen there will be stromogen. Study of arrangement of setae is also known as ketotaxy. And multicellular structures, there will be spur which are mobile in nature and spine which are immovable in nature. So we can, this is a representation of the non-cellular and cellular cuticular appendages. No epidermal association, rigidly attached, except minute hairs and thorns, and cellular epidermal association is there. Unicellular already we have seen, and the multicellular also we have seen. And coming to the clothing hairs, that is in bees, we can see the clothing hairs. Then in the bristles, which are seen in flies, and scales, which are seen moths and butterflies. So the first one here we can see the setae. The setae, you can see it is the setae is formed from the epidermal cell, which is called as the trichogen, and the surrounding cells are called as the chromogen. You can see here, you can see the base, the basement membrane, epidermis, then the pitch, the socket in which the setae is arranged. And coming to the scale also, we have the Lepidopterans in moths and butterflies. We can see the scale and how the scales are formed. It's also formed by a trichogen only. And coming to the glandular setae, where we can see the gland cell. These are poison setae of caterpillars, which will emit poison. In the case of many Lepidopteran caterpillars. So glandular setae, it is glandular setae. Then first one is the setae, the diagram of setae, how the trichogen, the chromogen, everything is formed, where the nerve connection is also there, and then spur, which is mobile, and the mobile spur and the immobile spine also you can see here. Then molting, the next thing is molting. Insect cuticle is hard and forms unstretchable exoskeleton, and it must be shed from time to time to permit the insects to increase their size during growth. Before the old cuticle is shed, new one has to be formed underneath it. This process is called molting. Whenever the insect grows, the old cuticle has to shed to increase the growth of the insect and the formation of new cuticle. The process is called molting. The periodic process of shedding the old cuticle accompanied by formation of new cuticle is known as molting, and is also called ecdysis. The process. The cuticular parts discarded during the process. Some cuticular part is discarded out. The older one is called exuva. The new instar comes out, bringing its head followed by thorax, abdomen, and appendages. Molting occurs many times in insect during the immature stages before attaining the adulthood. The time interval between two subsequent molting is called stadium, and the form assumed by the insect in any stadium is called as instar. Molting is a complex process which involves apolysis, ecdysis, and sclerotization. Apolysis is apo means formation, lysis means dissolution. The dissolution of the old cuticle and formation of new cuticle is known as apolysis. The process of formation of new cuticle by lysis of the old cuticle. Apolysis starts with repeated mitotic divisions of epidermal cells result in increase in number and size of the epidermis, which becomes columnar in shape and remain closely packed. The stage where the insect has both newly formed epi and procuticle and old exo and epicuticle is known as farrate instar. The agonizal membrane starting splitting along the line of weakness due to muscular activity of the inner developing insect and also because of swallowing of air and water resulting in distension of the gut. The breaking of the ecdysoid membrane is also due to pumping of blood from abdomen to thorax through muscular activity. After the breakage of the old cuticle, which is known as exua, the new instar comes out bringing its head followed by thorax, abdomen and appendages. So how the process of ecdysis. And next one is sclerotization. After shedding the old cuticle, a new cuticle which is soft, milky white colored comes out, become dark. The process of sclerotization, hardening through the process known as tanning. The change in color is called tanning and the toughening is called sclerotization. The tanning or sclerotization, both the processes were change in color and sclerotization. The process of hardening involves development of cross links between protein chains is known as sclerotization. The process of formation sclerotizing of tanning is called as sclerotization. This tanning involves the differentiation of the procuticle into outer hard exocuticle and inner soft endocuticle. 3 types of hormones are involved. Then steps in molting. What are the different steps? One is the behavioral changes. The larva stops feeding and become inactive. Changes in epidermis. In epidermis, cell size, its activity, protein content and enzyme level increases. Cells divide mitotically and increases the tension which results in loosening of the cells of the cuticle. Apolysis, the detachment of the cuticle from epidermis. The formation of subcuticular space, secretion of molting gel in subcuticular space which is rich with chitinase and protease. New epicuticle formation. Cuticle layer is laid over the epidermis. Then procuticle formation. Procuticle is formed below the epicuticle. Activation of the molting gel. Molting gel is converted into molting fluid rich in enzymes that activates endocuticle digestion and absorption. Then wax layer formation. Wax thread of the pore canal secretes wax layer. Cement layer formation. Dermal gland secretes cement layer. Pectocuticle. Then molting involves 2 steps. One is the rupturing of old cuticle and removal of the old cuticle. Coming to the rupture of the old cuticle, insect increases body volume through intake of air and water. It enhances the body blood flow to head and thorax. Thereby, old cuticle ruptures along predetermined line of weakness known as adaptable line. Removal of old cuticle. Peristaltic movement of the body and lubricant action of the molting fluid helps in removal of old cuticle. During each molting, the cuticular coverage of body, legs, internal lining of forehead, hind and trachea are discarded. Formation of exocuticle. The upper layer of procuticle develops as exocuticle through the addition of proteins and tanning by phenolic substances. Next one is the formation of endocuticle. The lower layer of procuticle develops as endocuticle through addition of chitin and protein. This layer increases in thickness. Then control of molting. The molting is controlled by endocrine glands like the prothoracic gland which secretes molting hormone. Endocrine glands are activated by prothoracic trophic hormone produced by neurosecretory cell of the brain. The hormones are involved in process of molting. Endocrine glands like prothoracic glands secrete molting hormone. Endocrine glands are activated by prothoracic trophic hormone produced by neurosecretory cells of the brain.